Good day, everyone. My name is Aslanach. This is my third year. My professor is Liu Jinghong professor. This semester is time scheduled. I did some of the experiments, also passed the qualification exam, and I attached my progress report checklist in my PPT, and my working title is the synergistic antitumor effect of the scavenger receptor LOX1 inhibitor in oral squamous cell carcinoma through the NF-kappa signaling pathway. Background shown that oral cancer statistics, and 90% of the oral cancers are the oral squamous cell carcinoma, and then new cases and dates is indicated increasing by the every year. And risk factor is increasing by the alcohol and metal, nuts, and cigarettes. And in oral cancer, LOX1 is highly expressed in oral cancer. And then LOX1 is that the lectin-like oxidizer LDL receptor 1, and that one is binding with the LOX1 into the cytoplasm, and they activated the NF-kappa pathway, and then ACT and ROS and inflammation some of signaling pathways too. And this background shown that one of the inhibitor IgG9 and simvastatin is inhibit that LOX1 expression and activated that the ERK and NF-kappa signaling pathway to the cytoplasm and nucleic. Also, NF-kappa is two different pathways, and canonical and non-canonical. We will focus that NF-kappa canonical pathway. The signaling receptor is binding the cytokine and toll-like receptor cells to the cytoplasm and directly interact with the IKK alpha and beta. And IKK alpha beta is interested to the IKB alpha. And when IKB alpha is degraded and P50 and P65 is released to enter to the nucleic. So in my experiments, shown that experimental design shown that I will check the how the cell proliferation is, and then checking the different expression gene by the RNA sequencing analysis confirmed by the qPCR Western blot analysis. And our expression of our LOX1, the target knockdown, that how is the cell proliferation is and how is the NF-kappa signaling pathway. We will prove that experiments also, which pass in vitro analysis. And also migration and innovation assay. Specific aims shown that the first aim is rule of the LOX1 in oral squamous skin cancer by cell proliferation and LOX1 silencing, and checking that the sum of the LOX1 inhibitors, simvastatin, IgG9, and aim to indicate that simvastatin inhibit the LOX1 expression and suppress the cell proliferation. And third, to determine the major NF-kappa signaling pathway in oral squamous skin cancer, cell proliferation inhibited by the simvastatin and silencing the LOX1 expression. Results shown that RNA sequencing and HSC, OCM, and SAS3 different oral squamous skin cancer cells comparing that the HSK without any treatment is highly expressed at 88 of regulation of the genes, and from there an OLR1 gene is high expressed in 3 different cell lines. And Kaplan-Meier and TCGA analysis shown that the survival rate of the highly expressed OLR1 expression, and it is they significantly increased that the survival rate. So we checking that the LOX1 the expression by the Western blot analysis, 2 different cell lines in here, and normalized data shown that. And after we will check that the time dependent, the silencing the LOX1 and time dependent and dose dependent experiments shown that the 72 hours experiments is significantly decreased the cell proliferation. This concentration from the literature, and we also confirmed that the Western blot analysis and 100 nanometer concentration and 72 hours is significantly knocking down the LOX1, the protein, and HSC3 analysis was shown. And we also checking that the how effect of the BI did this commercial LOX1 inhibitor, comparing that the 3 different cell lines. HSC3 cell lines is comparing other 2 is not too significant, higher expression than the others. The gene expression, mRNA expression that after the silencing LOX1 by the RT-qPCR analysis, 72 hours the results of 3 different cell lines with significant results, especially HST3 cell lines. Also, in kappa is signaling pathway by the RT-PCR analysis, but thus the cell lines is significant activate in a kappa pathway. And we will confirm by the Western blot analysis too. And OCM1 cell lines and normalized data shown that the phosphorylated and kappa pathway is decreasing. The SAS group is phosphorylated in kappa is significantly increasing. It is same like the RT-PCR analysis. HSC3 analysis shown they not too much activated the expression in is 100 nanomole concentration. Cell cycle analysis shown that the GO1 cell line the path is significantly decreasing after the silencing the NOX1, and this result also shown that the SAS cell lines HSC3 this cell lines is not too much differences like the OECM1 and SAS cell lines, and we also confirmed that the cell did by the flow cytometry, and then this result they shown that the early apoptosis is a little high, but the late apoptosis is significantly increasing the silencing the LOX1 with the vaccine, and we confirmed the apoptosis HSC3 also. That's shown. So we will confirm it until this experiment that half-simvastatin and commercial inhibitors highly decreased the cell proliferation in oral squamous skin cancer cell lines. And second, and simvastatin inhibited more effective in the this one